Feeling good, wanna feel great. Longevity starts, don't hesitate. Too much noise, what do you choose? Trendy hacks, conflicting news. Skip the myths, come hear what's true. From world-class experts straight to you. First and third Thursdays, 7 p.m. Science, health, and our team. Brain to heart, from food to rest. Live the talk on living your best. At 7, we'll see you there.
How can we actually go from knowing the genome towards operationalizing it and bringing it into interventions?
It's a great question. So we're working on multiple streams there, and I know that the Abu Dhabi government has big plans as far as how to use this in a very practical way to help improve the health of the population. And partly what we can think about is what can we do outside the health system and what can we do inside the health system. And so what the Institute is doing is really thinking about how do we utilize these data in a way that help us prove the model of care. So we are doing a randomized clinical trial of within the public health system to look at the model of care.
Welcome to the National University of Singapore Yonglou Lin School of Medicine Healthy Longevity webinar. Thank you for joining us. You just saw a fragment of our last episode talking about the genome with Dr. Nicole Sirotin, and she also talked about how to bring healthy longevity medicine into clinical practice. So watch it. You can find it on our YouTube channel. Today, we are talking about a different topic. Associate Professor Lingyan Shi is with us, and you will hear about vibration of molecules seen through the microscope. Very interesting. So please send all your questions using the Q&A function. Kamil Pabis, our PhD student, will moderate your questions, and he will introduce himself first.
Hello.
Hi there.
Can you tell me a bit about yourself?
I'm Camille. I'm a final year PhD student at the Center for Healthy Longevity. I work with Professor Brian Kennedy.
That's great. And so what do you work on as a graduate researcher?
It's really a wide range of topics. So it ranges from animal work over bioinformatics to human work. If you want, I can mention a few specifics. So we're very interested in combining treatments, supplements or drugs to mitigate aging. We're also very interested in important controversial questions in the field, like what works, what doesn't work.
So what is a very controversial opinion that you think is true, but most people don't know yet?
So one thing that we started looking at is, for example, exercise for longevity. We know it's very important, but on the other hand, it's also that the benefits are slightly overrated. I personally started to think that you need to really combine many different things, whether it's lifestyle, supplements. In the future, we will have longevity drugs and other things. We need to be much more holistic about aging. I think that's still somewhat of a controversial opinion.
That's very true. Health is very holistic after all. Then, do you exercise, or what other things do you keep yourself healthy?
Yeah, I actually exercise 4 to 5 times per week. I pay a lot of attention to good sleep, not to stress too much, and I do take a few supplements. I go to the doctor to check my blood work, and if anything's wrong, I would take drugs for that. And healthy nutrition, I pay a lot of attention to that as well. So I really try to have a mix of things.
Good for you. So why did you become an aging researcher, and who inspired you?
So just out of high school, I read a book by Aubrey de Grey called Ending Aging. So that he had the long-term dream that one day we'll be able to stop aging, and it really inspired me to work towards a better future. And I think even any small step that we can take right now to slow aging, as it turns out, this would have massive economic benefits, and that's why I fell in love with the field.
That's really nice. Would you want to live forever?
I would certainly like to live for a very long time, to travel all the countries, perhaps travel all the planets in our solar system, see what else is out there in the universe. It's a very big place, and there's so much to explore.
Which planet would you like to go first?
That's a good question. I always liked Pluto, although I think these days it's not considered a planet anymore.
Okay. And what is your favorite long-lived animal or living being, since we're on the topic of longevity?
Yeah, actually, it's more of a living being than an animal. I like it's a kind of tree called quaking aspen. They're like a clonal colony, so in a way they're immortal. There are some of these colonies that are up to 100,000 years old.
That's really interesting. And just a little bit about yourself personally. When did you arrive in Singapore?
It was almost 4 years ago.
And what was it like for you?
It was a bit hot, and I arrived just about after COVID, so there was not much going on. And together with a friend, I had this idea that we should start a meetup because there was not much happening in the longevity space.
So tell me more about this meetup.
It's called the Longevity and Health Meetup in Singapore. We're on meetup.com, and these days we do monthly meetings. We invite experts from NUS, from industry. We provide free food. We do networking. It's a lot of fun.
That's really great. And what kind of other projects or groups are you involved in?
Recently, we're also trying to start a student group called the Healthy Longevity Initiative with one of my students. So if you ever want to get involved in any of the projects, feel free to reach out to me or to us.
How can we follow you for more?
One way is for Twitter. I'm the aging scientist there, or also on Substack on the Geroscience blog.
Awesome. And my final question is, what would you really like to do for yourself today?
Today, well, I have a lot of meetings, so I'll probably need to get a coffee first.
All right. Thank you so much, Camille. We'll see you soon.
Thank you, Camille, for this nice introduction. And now I know that you are also drinking coffee. So let's switch the topics. Our guest today is Associate Professor Lingyan Shi from the University of California, San Diego. It's very early for her, so thank you so much already to be here. With a background in molecular imaging and postdoctoral training at Columbia University, her research focusing on the developing label-free multimodal nanoscopy to visualize cellular metabolic activities in aging and, of course, also disease. Her pioneering contribution includes discovering the optimal golden window for deep tissue imaging and advancing stimulated Raman scattering microscopy into super resolution nanoscopy. Like, I am super fascinated about all the words, what the golden window is and what a Raman microscopy is and the scattering of it. So Lingyan, the floor is yours. Thank you so much to make time and in this early phase of your day today. So thank you so much.
Thank you so much for the great introduction. And I'm going to share my screen. So hello, everyone. I am so happy and very honored to be invited to present for today's seminar. So my title here, Visualizing Metabolism in Aging with Multimodal Nanoscopy. And from the introduction, we already heard about the nanoscopy. I'm going to talk about it today. But before that, as a biologist or medical researchers, our dream is to visualize the endogenous molecule inside the tissue or cell without any labeling or staining. So look at this label-free imaging. So this is a totally label-free or staining-free image from a mouse brain cross-sectional area. And you can see different colors, the green color, the blue color, and you can see there's a very intensive blue color here with my mouse pointing. That is protein signal. Protein signal is in blue color, and the lipid signal is in green color. So the blue and the green color are protein and the lipids. They can be visualized directly without any labeling or staining from this image technology. And if we enlarge or zoom in to look into the detail inside that tumor, this is actually a a brain tumor growing inside the brain. So you can also see some extracellular matrix such as collagen in this magenta color here. So the collagen network can be very different in the center core of the tumor towards the boundary of the tumor. It's growing inside the brain tissue, and it's very different in molecular composition compared with the brain tissue. Or from the human biopsy tissue, you can also visualize different type of molecule without any labeling or staining, such as kidney biopsy tissue. So the kidney biopsy tissue showing here, you can visualize in different color channel these magenta color. Magenta color again is a collagen, and the blue color dots on top are lipids droplets. These blue color in the center, or green color or red color in the center, they are from protein, different protein composition from different subtype of cells inside the glomeruli, and also ducts. You can see very big dots. They are accumulated lipid droplets. So it's like a dream that we can visualize these different types of cells or molecular compositions, or without any worry of labeling or staining. Then you can also do quantitative study from a top layer to the bottom layer. For example, you can see the multiple color image layers by layers and figure out the molecular composition heterogeneity from the group glomeruli towards the tubule side in this tumor, this kidney tissue. So that can be taken by this image called label-free nanoscopy, which is a multiple modality integrated together. It's built in the lab, in my lab at UCSD, and you can see it integrates SRS, stimulated Raman scattering, and MPF, multiphoton fluorescence, and fluorescence lifetime and second harmonic generation. So you will see that if you place the sample, such as cell or tissue or animal, under the microscopy objective lens, you can collect different type of species of molecule all at once in the same region of interest or in the same volume. And then you really can do the quantitative analysis from these different modalities of signal. So as we just mentioned, the human biopsy tissue can be visualized directly without any labeling. So label-free biopsy tissue or other cultured cell or animal, even in live animal, you can visualize them directly. So you actually visualize with fluorescence at the same time, and the fluorescence microscopy is very familiar for everybody here. It's very high spatial resolution. Also, a lot of commercially available fluorophore that you can easily purchase and target the protein molecule you want to visualize. However, it doesn't really label many of the lipids for fluorescence microscopy. In contrast, the Raman scattering that I want to introduce specifically here is a very particular vibrational imaging technology. So Raman scattering, it was discovered first by C.V. Raman, an Indian physicist. He won Nobel Prize in Physics in 1930. So when the photon interact, or you can say you just shine light on the biomolecule, biological tissue, those molecules inside the tissue or cell will absorb the energy from the photon, and the chemical bond inside these molecules starts to vibrate at a certain frequency. So the vibrational frequency can be detected by this Raman spectrum on the lower right side. If you look into these spectrum peak, those peaks represent different chemical bond from the molecule. So CH is like carbon-hydrogen chemical bond. If you want to visualize them, in theory, you just pick up that peak and do the image to map out how these chemical bond looks like in the XY spatially, or even in the volumetric way. However, the spontaneous Raman is very low in sensitivity. That's a problem. In the past decades, people were working so hard of enhancing the sensitivity for this Raman scattering technology. Not until 14 years ago, when Dr. Sunny Shi and his fellows in Harvard University, they invented the new type of microscopy called SRS, stimulated Raman scattering. So this microscopy is different from spontaneous Raman. This we just showed in the previous slides. Instead of utilizing one laser beam in the spontaneous Raman microscopy, the SRS microscopy utilizes two laser beams, the pump and the Stokes. And the energy differences between the two laser beams can match the vibrational modes of the molecule of interest. At that moment, you will see that the signal or the transition can be dramatically enhanced by 10 to the 8 magnitude. So the signal level can be really increased dramatically with this joint effort from two laser beam. With this technology, we can easily take image from those chemical bond inside the molecule. So you can see that needle biopsy, it's a kidney needle biopsy from human body. The patient's worried about what kind of molecular changes happen in these kidney tissue. Then you can just directly visualize this kidney biopsy tissue. It's like a needle tip. You can see from this tip to that, and you will see different color. So these different color represents different molecular composition, because each pixel on the image will have that Raman spectrum profile. So the Raman spectrum on each pixel contain different molecular composition information, and you can dissect them based on the molecular signature or fingerprint in the Raman spectrum. You can look at the right side. If you look into the saturated fat, unsaturated fat, these are from top to the bottom, a few different species of molecule that you can detect already. So saturated fat, unsaturated fat have very different signal because of the carbon-carbon double bonds from the unsaturated fat. Protein, lipids, they are very distinguishable with different Raman peak position. Collagen, flavin, and DPH can be also detected by other modalities such as second harmonic generation or fluorescence. So if you zoom into those regions of interest and you want to investigate further at the subcellular level or subcellular organelle level metabolic heterogeneity or the molecular composition, you can see region 1, 2, 3, 4, and those are really looking to single cell level or subcellular organelle level and dissect the molecular composition. You can also do, for example, 3D from top to the bottom layer. You can do 3D reconstruction to visualize how the lipids or protein located in each cell in the piece of tissue, and that's 3D volumetric image from tissue, the mouse skin tissue. So you can also do video recording, and you can even look at where is the newly synthesized molecule and how much they synthesize the new molecule after a certain time duration. So this is a video recording. You can see blood is still flow here. The mouse is still alive and under anesthesia. And you can do 3D to visualize how those molecules are located in each place. So really, I think everybody will be excited about this colorful image, and you can see different species of molecule already, but there are still major challenges such as these 4. I just put molecular specificity, newly synthesized from pre-existing molecule, super resolution to see organelle level, co-registration of multimodality. So these are the challenges that I'm going to talk about one by one and how we overcome these challenges in the past few years at UCSD. Each molecule has its own Raman spectrum profile, as we just mentioned. So if you see this spectrum library, it's a library of spectrum that we measure from different molecule. So each molecule is like a 100% purified molecule. A molecule can manifest itself in multiple peaks. So you can consider it is like a fingerprint for each molecule. They have their own Raman spectrum shape. They have own peak position Each peak have their own full width half maximum in different location. So at the first glance, they look similar, but if you look into these details, the feature can be really utilized for distinguish each type of molecule. And you can also consider molecule they speak at their own voice. So their sound wave are different from each other. So although in one pixel of the image can have mixture of different molecules, but you can still detect them based on their own Raman spectrum profile. That's the method we developed here. From the panel A, B, C, D, you can see from panel A, it's the library that we measure the purified compounds and generate the library. Panel B is we are imaging that tissue or cell or animal in the hyperspectrum stack way, so you can have a stack of image to generate the panel C spectrum profile for each pixel. And how we can detect, like panel D is a detection of each type of molecules. There are subtypes of lipids molecules such as sphingosine or sphingolipids, high cholesterol or high TAG triglycerides. So high means very high scored. So you detect in that way that you do the reference matching with panel D for each pixel, and if the score is very high, then you can assign that pixel have a dominated amount of, for example, sphingosine here in the red color pixel, and those green color pixel have a dominant amount of cholesterol molecule, and the blue color dominated by the triglycerides molecule. So if you merge them together over the three-color image, this three-color image is from the human brain tissue slides. And it's the first time we detect subtype of lipids in a piece of tissue label-free. Then going into some detail, for example, you want to really see single cell level or very, very detailed subcellular organelle level, you can really look into those human brain tissue sample. It's control aging brain that you can see a large amount of sphingosine here signal in the magenta color, and then the cardiolipin signal here in green color, and you can zoom in to analyze a single cell level metabolic heterogeneity or molecular composition heterogeneity here. But if you don't want to see a single pixel for pixel by pixel level, you want to see cellular organelles such as lysosome. We want to detect lysosome or lipid droplets, then how we can detect them without any labeling. That can be also done for bringing in a concept of superpixel. So you can see the lysosome have a bunch of pixels all together here in the cyan color, and the lipid droplets in magenta color, and other organelles with a lot of PE, those phospholipids, and those are in green color. I don't know what is this organelle, but we recognize the lysosome in cyan color, and the magenta color are lipid droplets. The way that we detect is, first you measure the lipid droplet spectrum and put it in the library as a reference, as a standard spectrum. And also you measure these lysosome in lysosome, in cyan color lysosome, and put these standard spectrum in the library. Then you can detect in a large scale piece of tissue where are lysosome located, where are the lipid droplets located based on the similarity score for these superpixel concept. So how you can validate they are real, for example, real cardiolipin molecule that we just showed. The cardiolipin can be also stained with an NAO fluorescence staining. So you can validate it with the fluorescence microscopy to stain these cardiolipin signal. So the 2-photon fluorescence show this is the cardiolipin located. And we did the detection on top of the same cell, the same bunch of cell. We see the label-free detection with this SRS technology, stimulated Raman scattering. And when we knock down this enzyme, so the gene knockdown can reduce that cardiolipin synthesis. For example, SHPGS1 is one of the enzymes that we can knock down, in the cell. So when we knock down the NAL stand cardiolipin signal reduced in the fluorescence channel, simultaneously the SRS detection of these cardiolipin also reduced. So it's truly detecting this cardiolipin signal. That validated, and we utilize this technology to detect, for example, aging mouse brain. So the mouse brain become old. When the mouse brain become old, we look at the phospholipids level and the cholesterol level in a subcellular resolution image. So you can see from the figure, there is a increase the free cholesterol accumulated in a certain group of cell. Not all of them, but some of the cells have elevated amount of cholesterol. But in many cases, we don't know what molecule we want to look at, and we have no idea what type of molecule this piece of tissue has. For example, the human brain tissue, human lymph nodes, human kidney, or even those Drosophila fatty body or mouse brain or Drosophila brain. If we don't have a targeted molecule to detect, you can still do label-free unsupervised clustering. So these different type of molecule or different type of cell with different molecular composition, you can see different color can be actually separated from this label-free hyperspectral image. So let's consider human brain as an example. There are a yellow color cell or green color cell. These are different type of cell with different molecular composition. We don't know what exactly the cell are, but they are different subtype of neuron, and that can be validated by using other approach. For example, you can do codex on top of it. You can do Xenium. So that is really cool that you still can see that different type of cell or different molecular composition immediately with this label-free technology. And I specifically want to look into a single cell that is dividing. It is doing mitosis, but not separated yet. At that moment, I did this snapshot to look at those newly synthesized DNA. So I'm telling you, this red color pixels are newly synthesized DNA, and other molecule in other different type of color, different colors as representing different molecular composition. But why this is newly synthesized DNA, not pre-existing old DNA? That is how we can visualize the newly synthesized and distinguish it from the pre-existing molecule based on this dual-size technology. So the dual-size was utilizing the heavy water as a universal metabolic probe. That means the heavy water molecule have two deuterium and D2O. That molecule have two deuterium atom, and one of them can be ripped off and attached to those small metabolites first. For example, acetyl-CoA amino acids, ribose, deoxyribose. You can see fast attachment or it's a enzymatic incorporation. You can slowly incorporate these delabeled small metabolites into macromolecules such as lipids, protein, DNA, RNA, or carbohydrates. And there's a new chemical bond formed to replace CH. CH is carbon hydrogen. And after the incorporation and synthesize into a new molecule, there is a new chemical bond, carbon deuterium. Why the carbon deuterium molecule can be so, carbon deuterium chemical bond can be so important? So if you recall all the Raman spectrum I showed in the past or previous slides, you will see the spectrum have a pattern. That is the natural biological samples spectrum pattern. No matter you measure them from plants or animal cell, they all have this pattern. You have signal on the left side, signal on the right side, but there's no signal in the middle from 1800 centimeter inverse to 2800 centimeter inverse. There's no signal until you have a new chemical bond formed like CD, carbon deuterium, inside those newly synthesized molecule. This means you can simply pick up the CD peak signal and image to map out where are the newly synthesized protein, lipids, DNA, RNA, or carbohydrates located at subcellular resolution. So with this technology, we can look at dual-size image of newly synthesized, for example, magenta color of DNA, or new protein is in blue color, new lipids is in green color. So if you merge them together to form a multiple color image, you can visualize many different type of newly synthesized molecule at subcellular resolution. And yet, this image methods can be applied to, for example, identify the tumor boundary in the glioblastoma tumor case. When I inject some tumor cell into the mouse brain and grow into a solid tumor, and let the mouse drink a few days of actually not very long time, maybe 1 day or 2, you can visualize these tumor cells which have a higher metabolic activity or turnover rate is really higher than the surrounding brain tissue. So the tumor boundary can be easily identified here. So this case can be applied on human patients. If the surgeon cannot identify those tumor boundaries with naked eye, they can let the patient drink some heavy water, and then you can see that metabolic activity differences between the tumor and the normal supporting tissues. Then they can be removed entirely. And we applied this technology to study the tau overexpressed mouse brain. So this tau overexpressed mouse brain project showing that when the tau protein is overexpressed in this mouse brain region, you can easily visualize the newly formed lipid droplets accumulation. So those lipids will be accumulated when the tau protein is overexpressed in the brain. Not only this mouse model, we also did the human iPSC cell for these neuron and the microglia cell. They also have this phenomenon when the tau protein is overexpressed, a lot more newly synthesized lipid droplets accumulated in the neuron and microglia cell, especially the microglia cell. Microglia cell accumulate a large amount of newly synthesized lipid droplets, and the function of the microglia cell will be perturbated. So it's like become dysfunctional after accumulation. And that phenomenon can be rescued by activation of AMPK. The AMPK activation can largely reduce that accumulated lipid droplets in microglia and restore the function of the microglia cells, which is very exciting for us to visualize that. And we did some diet change, for example, with different diet. In animal model, we already know eat less or diet restriction can prolong animals' lifespan, but nobody really look into the brain, for example, brain lipids metabolism or metabolic change in the body, how the metabolic activity has been changed after you eat different type of diets. So STDRHGHS here is from standard food to diet restriction and high sugar, high glucose, high sucrose, high fructose. So there are different type of sugar and in high level. And we give this 5 different type of diets to female and the male animal. So this is Drosophila animal. In the female group and the male group, we both give them these 5 types of diets and look at the metabolic activity change. So when the Drosophila become old, for example, the female Drosophila brain lipids have an elevated metabolic turnover rate, especially the lipid droplets. Lipid droplets have a metabolic turnover rate increased in the diet restriction, under the diet restriction. But if we give more sugar, you can see they have reduced. It's actually not that high anymore. High sugar, high sucrose is even lower. But we look at the male group here, the same age, older male, they have very different phenomenon. So we observed that the diet restriction also increase the metabolic turnover rate in the brain lipid droplets. But when you give more sugar, no matter you give glucose, sucrose, or fructose, they stay strong. So this is very interesting results. We see that female and the male brain, when they age, they have very different sugar tolerance. It shows that the female are more sensitive when they grow older, more sensitive to the sugar. So sugar change lipids metabolic activities. But for the older male, those animals shows not that sensitive to sugar. So perhaps as a female, we should be careful about sugar when we grow older. But male, you can feel free and eat. We also want to mention many different metabolites that can be deuterated. For example, we can deuterate the glucose. So deuterium can be labeling the glucose molecule, amino acids, fatty acids, choline acetate, or even neurotransmitters. So this is a metabolic metro map showing the different metabolic pathway, and you can do rewiring with different metabolites simultaneously, actually. So this is cool. Instead of using the D2O heavy water, you can also add other. You can add deuterated glucose, deuterated amino acids, fatty acids. So whatever you do, these deuterium eventually will be incorporated into the final product, like the biomass newly synthesized. For example, the glutamine, if we do the deuterium labeled glutamine, the glutamine will be utilized as a building block to generate new protein, lipids, DNA, RNA. Those type of molecule can be distinguished based on the Raman spectrum shape differences. So although they are all derived from glutamine, but you can see the protein, lipids, DNA, RNA, they are newly synthesized with different Raman spectrum shape and peak position that can be distinguished. So we apply this with different metabolites, and this is a new technology called the Summit SRS. We do multiple metabolites. For example, you throw in glutamine, glucose, methionine, tyrosine, leucine, or even more other type of metabolites. You can either give the cell culture media that kind of molecule. You can also let the animal eat, or let the human eat this kind of molecule. Eventually, these molecule will be incorporated into macromolecules such as protein, or you can call newly synthesized protein. So this new protein derived from D metabolites can be visualized and quantified at subcellular resolution. So this is a comparison study for spheroids, like more natural spheroid, tumor spheroids. It's a breast cancer spheroids. So the network is more like aggressive. So they are more aggressive network spheroids. Compare with two different phenotypical expression, you will see that the amount of newly synthesized protein is more significant in the network. So they are not glutamine, but not glutamine or glucose, but methionine, tyrosine, or leucine. They have an elevated amount of protein synthesis. So it is indicating a metabolic pathway shift in this more aggressive network of these breast spheroids. And in the lipids part, you can also visualize the methionine is more into newly synthesized lipids here, but not in the control spheroids, but the more aggressive more aggressive network spheroids breast cancer, you have larger amount of methionine derived lipids here that can be really visualized and quantified. It's so interesting. And if you want to really look into the organelle level, for example, which organelle have what kind of newly synthesized molecule. So this is one case that if you see the glucose-derived lipids in the lipid droplets, and the deuterium labeled methionine-derived lipids is located in the lysosome. So with lysotracker, in the end, you can see they have very different carbon source, or the building block derived new lipids. They are all new lipids, but the location of the newly synthesized lipids is different in the cellular organelle. So magenta color is D-glucose-derived lipids. They mainly go into goes into these lipid droplets and stored there. But the demethionine derived lipids is mainly going to the lysosome. So this is from the late stage larva of the Drosophila. It's actually a very age-dependent phenomenon that you can observe. So as organelle level, we need to develop these methods called apod SIs to be able to see a single lipid droplets level metabolic heterogeneity. And we understand a single cell is the basic unit of life. Those organelles such as mitochondria, lipid droplets, lysosome, Golgi apparatus, or ER membrane, or even the nuclear envelope, these can be super important, especially those molecular flux among these organelles, and those molecular composition change in this cellular organelle can be super important to visualize and quantify. So we developed this apod SIs. This method is going to tell you how we can do deconvolution to achieve that spatial resolution. So you can consider many pixels in the image as a massive amount of emitters. And that massive amount of emitters can simplify this question toward finding out the minimized distance between the image and the point spread function. Then you can relocalize them to be more precisely localize this massive amount of emitter to generate the super resolution image, such as a human retinal rod cell, which shows in the yellow box here. On top, it's without super resolution, and on the bottom is with super resolution. You can see layers of lipids membrane layer. So these layers of membrane, they have very different lipids composition in the aging processes. So when the rod cell is aged, you can see outside the boundary layer towards the core center of that membrane has a lipids composition change. Even the lipid saturation can be also different. And if you look into those cellular organelles, such as magenta color mitochondria in this magenta color box, magenta color is a mitochondria, and the surrounding yellow dots, they are SS image of lipid droplets, and the cyan color are protein-enriched organelle. They interact with each other, and you can see that so clearly under the super resolution approach. When the cell is under stress, you can see the yellow box. When they are stressed, no matter it's hypoxia stress or it's other environmental stress, nutritional stress, you will observe more accumulated yellow color lipid droplets. So this label-free lipid droplets signal will allow you to visualize and quantify how much of the newly formed lipids are accumulated in the surrounding area of mitochondria and how they interact with these other organelles in the surrounding area. These lipid droplets can have different lipids composition inside. So at a single cell, you can see the single cell level. If you want to see whether these lipid droplets have different lipid composition inside each of them, we can really detect the molecular composition based on the Raman spectrum shape and peak position and those features that we mentioned before. And then the different color represent different composition of the lipid droplets. Some of them have more cholesterol. Some of them may be dominated by triglycerides. Some of them may have more equal amount of both. So they can be really looking into even single lipid droplets level. When you give more sugar, for example, 3 times of glucose treatment, this is from the Drosophila fat body lipid droplets. So the animal eat more sugar and you can see more lipids, new lipids synthesized in that single lipid droplets. These are more cyan color that wrapped in the surrounding area of lipid droplets. They are newly synthesized, but the red color are old lipids droplets. Do I have more time? Maybe I want to, so let me see. Okay, so we will have a few slides to end my talk for today, the correlational multimodality, because we just mentioned if we don't know what kind of cell we are looking at, we can do other technology or other methods to co-register and then identify, for example, codex or xenium or even fish. So how we can incorporate other technology. We showed some example here with light sheet, 3D volumetric image that those volume of the tissue from the human biopsy, human kidney biopsy, we can visualize those in the, for example, the collagen network, how they wiring together with those glomeruli and the nerve in this light sheet sample. And from that piece of tissue, we can also identify those different layers of glomeruli from cortex layer towards the medulla layer. What is the lipids subtype or lipids composition change from different layers of the glomeruli? So you can detect the lipid subtype, and you can compare the aged, for example, during the aging processes, how the layers of cortex towards the medulla layers of glomeruli have changed the lipids composition, how the cholesterol level has been changed at subcellular resolution. We can also co-register with codex here. So codex can tell us what exactly the cell type is. So on the left side is from the codex done from Professor Rongfan at Yale University. They did that codex on the human lymphoma case. So you will see that after the metabolic activity imaging, it's a label-free multimodal approach we did. After that, they did the codex on top of it. And to co-register on this particular bunch of cell, we can zoom in to a specific cell type, for example, large B cell here. If you look into this large B cell in the lymphoma case, they have a lot more lipids accumulation on this nuclear envelope. And you can quantify that lipids and also identify the saturation level. They are unsaturated or saturated. You can also look into very detailed analysis. And we also did the MALDI and SIS imaging of human lung in this Atlas, NIH Atlas. It's called HubMap, Human Atlas Program. So this program provides a lot of human patient sample, and we can look into that human tissue in different organs. And this is one example how we can really identify the molecular composition, both from the MALDI in larger scale and from our metabolic imaging, we call the label-free nanoscopy in the subcellular organelle, even those very little tiny small region of interest, and molecular composition can be identified with multiple scale because MALDI is more larger scale, and our approach is zoom in into cell. And that can be also combined with codex together to even identify more biomarker. So this is some example that I'm not going to get into the detail due to the time limitation. So to summarize, we talked about 4 different key technical leap here. PRMSIs, we talk about how we can detect diverse types of biomolecule with that Raman spectrum features. A part is about super resolution, how we can look into the cellular organelle, even different layers of membrane, lipids membrane in the rods or in small organelle level, like lipid droplets. Bioorthogonal SIs is mainly about the deuterium labeled molecule, so we can label multiple metabolites, glutamine, glucose, or amino acids, fatty acids, choline acetate. We can see that metabolic activity or turnover rate or anabolic activity. So this is really metabolic dynamics that we can visualize at the subcellular organelle level and co-register with other modalities such as codex, Daniel fish, MALDI, light sheet. We can really look into more angles, more molecular composition from different angles, right? So we have SIS, multiphoton harmonic generation and fluorescence lifetime combined with every thing or every approach together. That is a imaging approach or imaging platform for us to visualize molecular composition plus molecular metabolic dynamics at subcellular resolution. So that is good for study aging and diseases and metabolic rewiring. And I will end my talk here. And thank you so much for your attention. Also, I want to acknowledge my group members, collaborators outside of UCSD, collaborators at UCSD, and my funding source. And I'm very happy to answer your question if you have any.
Thank you, Lingyan. I haven't seen such a beautiful presentation with so much powerful images, so powerful, lots of Picassos, I would say, showcasing the molecules. Let me ask the first question. You were really nicely showing that you can distinguish proteins from lipids. Can you also look at the protein quality, for example? We know that folding processes with the aging process are going wrong, misfolded proteins. Tell us, is that the new avenue to really see how appropriate we are shaping our body?
Such an important point you just raised. So I show a lot of lipids in the slides because we have limited people in the lab to work on one of the molecule. Actually, protein is super interesting. So the Raman spectrum shape that I showed you in those like those noodle vibrating, right? That peak in the left side, it's called a fingerprint region. That amide region, particularly between those like 1650 region, these have secondary structure information from the protein molecule. So you just mentioned if the protein is folding or unfolding, or if the protein is doing some dynamics, that region of the Raman spectrum shape, peak position, each peak's full width half maximum can change after those movement inside the protein molecule. And that could be a very good PhD project if somebody is very interested in study that kind of dynamics and the signal change. I think this is so cool to visualize them even in real time.
So you're advertising at the moment. Very good. May I ask you, maybe I didn't hear it well, but do you say that you can even do live imaging with
Yes.
mice? Okay. So what does that mean, live imaging for humans? Could we, when I would give you my arm, could you actually live image my skin?
So skin is very accessible. Skin is the best part of the human organ. It's the largest organ, and it's actually a very important organ tissue for study aging processes. The light doesn't penetrate much into the deeper level of the organ. For example, if you want to see a liver, it's very hard because that penetration is limited for a few hundred micron. So the skin is perfect.
How deep can you get in?
So it's a few hundred micron. It's like
Okay.
a two-photon fluorescence. The depth of penetration is same as two-photon fluorescence because we use the same laser wavelength range. The golden window can make a little bit deeper. The golden window you mentioned in the beginning, it's from 1600 nanometer to 1870 nanometer window. That window can reduce the scattering in the tissue, and you can penetrate even deeper. So that possibly enhance the penetration depth, even in the human skin study. So in 2010, Dr. Sandy Shi published a paper on how we can look at that skin by making a video, right? You want to see different time points and see dynamics. So that is backscattered SRS. The image that I showed you today is forward transmission level of SRS. The signal will be higher, but if you do backscattered to look at those skin tissue metabolic dynamics, the signal level can be lower than the transmission, but still detectable. So yes, for skin, it's really a great technology that you detect the skin lipids metabolic change or protein metabolic change, or even those morphological dynamics combined with the molecular composition change.
Yeah. So you already said you can disentangle protein, lipids, and maybe even what kind of proteins in terms of quality they are. Let us zoom out a little bit, not zooming in, but zooming out. if you would have the skin, could you detect the macrophages or other cells, and could you detect if they would be senescent or not? So at what stage of differentiation or even senescence they are?
Yeah, so this is actually the ongoing project that we want to really identify, for example, which cell type, right? We know that the molecular composition is different in different subtype of cell or different types of cell. So macrophage, T cell, B cell, or whatever other skin, for example, other skin cells inside this image can have different molecular composition already. But when we do the hyperspectrum image and identify the cell type with different color, that doesn't really recognize what kind of cell they are. But fortunately, we have other technology which can be further, utilized to identify that cell type. So now in the AI era, with the machine learning or even the training with the help from AI, I think it's really doable that we have large number of sample to be trained, right? To really automatically recognizing these, so they can automatically be recognized by this training. After train that kind of model, you can see, for example, the macrophage always have certain molecular composition, and for example, they have a different types of molecular composition, but they have that range of freedom that you can really let the machine learn and recognize after certain period of learning process. So this is possible.
I have one more question before handing over to Kamil Pabis, who has many questions from the audience too. So we have to restrict it a little bit. But what fascinates you most in your work? Short answer.
Very exciting to see some of the metabolic rewiring phenomenon when we study, for example, the aging process in animal model. It's super cool to discover some real-time metabolic dynamics change with different, for example, building block or carbon source, and we will say, oh, we found something potentially can be the therapeutic target. So to restore some physiological condition with a certain molecular target, it's so cool.
Okay, super cool. Kamil, take over.
All right. Thank you, Dr. Shi, for the amazing presentation. You made a complex topic really quite accessible, I think. And maybe briefly, just 2 or 3 questions from the audience. So one person asked, what biomarkers can you see that other methods cannot see? Maybe you can compare to something people understand, like MRI, CT. How are you different?
Most of the biomarker that a protein molecule, people can see protein molecule very clearly by staining or fluorescence technology. But the protein, protein is that molecule that I think fluorescence can be really the best choice for now. But I'm not saying in the future. Lipids, I specifically pointing out lipids because it's very hard to be stained or to be labeled with other fluorophore. And even if you label that, that is not exactly labeling the lipids molecule. Usually you are labeling some protein molecule on the lipids membrane or somewhere. So the lipids molecule can be directly visualized at subcellular resolution. This is something unique with this Raman technology. So it's different from MALDI because MALDI, you don't have that subcellular, like a nanoscale resolution, and you cannot do live animal, live tissue. That part is different. However, it's complementary with the MALDI technology because MALDI really tells us very precise molecular information. So molecular detection can be cool in MALDI image. And that is like generating a complementary multi-scale image if we combine 2 technologies together. So SIS is going to visualize those subtypes of lipids at cellular organelle level on a 59 nanometer scale. So then that can be zoomed in detailed analysis. And for PET, you just mentioned PET. PET can visualize metabolic activity or dynamics in very large scale organ tissue, for example, the whole brain or the whole body even, or whole liver. So that part is really cool. And in live human body, this is something that the SS cannot do, but the SS can do detailed cellular level metabolic turnover rate. And another difference is the tracer or the contrast, the PET imaging utilizing analog of glucose, for example. So one of the analog of glucose, they can be uptaken by cell or those tumor or even those liver, no matter what kind of cell uptake them, they only uptake them. They don't metabolize them because the analog cannot be utilized as a real glucose anymore. They are not glucose. But in the SIS case here, you just look at the glucose with deuterium, and the deuterium labeled glucose can be utilized as a real glucose molecule. So you can see how these deuterium labeled glucose are metabolized as an energy source to generate ATP, or you can also see how the glucose molecule can be metabolized and anabolic metabolic pathway, and they can be synthesized into new protein, lipids, DNA, RNA, carbohydrates, and you can still visualize how they are incorporated into that new molecule and where they are located in the end product, the newly synthesized biomass. So these are very different.
All right, maybe very briefly, the last question was about live imaging. We saw some images of blood, like what is your frame rate resolution and what are the smallest events that you could see in the live?
So this is an important point for people who want to see live chemical reactions or molecular like flux in the cellular level. So for single cell, I'd say if you're only interested in one single cell or even a small cellular organelle, you can do fast. You can do really fast image, like millisecond frame. But if we look at thousands of cells in a relatively bigger field of view for those protein and the lipids, so normal protein of lipids that I showed you in the slides. So you can take the brain image for lipids like CH2 chemical bond, that bigger region of interest, it's about 500 micron multiplied by 500 micron. I think it's around that range. Takes like 2 seconds to image that field of view. But if you want to just cut into a single cell, that could be really faster. But for the CD signal, so the signal level matters. CD signal is a newly synthesized molecule, which is a small amount. The concentration of that CD labeled molecule is much lower than those original existing protein and lipids. So it's about 26 seconds for that bigger image that I showed you, like one frame, 512 pixel multiplied by 512 pixel level. So that's take like 20 seconds.
Good. Thank you so much, Lingyan. It was a wonderful presentation. I think we all hugely enjoyed the imaging, and thank you.
Thank you very much,
Camille, for really nicely presenting the questions. So we have a couple of updates. We will have a Healthy Longevity Intensive course and a conference end of February. The intensive course is at the 23rd to the 25th of February, and the conference at the 26th and the 27th. So if you would like to come to Singapore, or you're already in Singapore, please submit your abstract. There will be best poster prizes and presentation prizes. And if you're a startup, be part of the pitch competition and win the best pitch Prize. On the 6th of November, Assistant Professor Peter Litsky will come, and that's a very stimulating topic. He will talk about the programmed aging. Why do we have to die? Why does evolution kill? We will end this episode with a short teaser. As always, take care.
So my colleague Vanessa already showed our new clinical trial center. However, now it's ready. We also conduct other tests such as measurement of hand grip strength.